Good morning, everybody. Welcome to the RCN webinar. We are going to give it a minute here while everybody trickles in before we begin. We're going to start at 8:02 with the introductory slides. Okay, we're going to go ahead and get started with the introductory slides today. So welcome everybody to the webinar for December for the RCN. Today we are very happy to have two exciting talks. The first is going to be by Liang Zhao, who is in the Department of Civil and Environmental Engineering at MSU, talking about STIs, tracked in wastewater. And the other is Anthony Maresso and Eric Boerwinkle, who are from Baylor and UTHealth Houston School of Public Health, to talk about some of their work on sequencing viruses in wastewater. A couple announcements is that we do have the RCN Student Discord, and Tamina will put the link in the chat if you'd like to sign up for that. We have a journal club and an email list. If you're not on them, you can contact us and we can get you involved in those. And lastly, we started a participant certificate program for students and postdocs. So if you are interested in that, you can scan the QR code, or Tamina can provide the link in the chat. And finally, just a reminder that because of the new year, our January webinar is canceled. It's too close to January 1st, and most of us will still be, our institutions are closed. In February, we have one of our speakers confirmed from India on vector-borne pathogens, and we're still finalizing the second one. So we'll be in touch by email to remind you of all that. So with no further ado, I am going to stop sharing my screen and I'm going to pass it to Tamina to introduce our first speaker. Or Liang, you can actually probably just start going because I've already mentioned where you're from. So thank you.
Thank you. All right. Thank you, Dr. Bain, for the kind introduction. And good morning, everyone. My name is Liang Zhao. I'm a postdoctoral research associate at Michigan State University. I work with Professor Irene Zagorarki in Civil Environmental Engineering Department. First, I want to thank for giving me this wonderful opportunity to speak at RCN webinar about our research. And today I'm going to talk about the tracking sexually transmitted infections in the Detroit metro area by molecular analysis of environmental samples. My talk has three parts. In the first part, I will just briefly talk about the wastewater-based epidemiology research happened during the COVID-19 pandemic and post the COVID-19 pandemic in Detroit. Then I'm going to spend most of my time talking about tracking sexually transmitted infections using WBE. Then I'm going to summarize with some conclusions and future work. First, I want to take a brief minute to highlight the Tri-County Detroit partnership. This partnership started in 2017, and we have closely worked with our industry partners and local health department and water utilities in Michigan. And we have constantly meeting with each stakeholder and updating the data and receiving feedbacks from each stakeholder. So the research we have done, the outcomes of our research is truly a result of collaborations and frequent communications and a team effort. So here, this slide shows the projects that I mainly participate in Dr. Zagoraki's lab during the COVID-19 pandemic and post-COVID-19 pandemic. So during COVID, we developed advanced methods to predict temporal and spatial fluctuations of COVID-19 in Detroit based on WBE. And after COVID-19, we try to expand our methods beyond COVID to some other communicable diseases. First, we develop a ranking system to prioritize what could be the future targets. And then we focus on sexually transmitted infections and some other gastrointestinal infections. And today I will focus on tracking sexually transmitted infections in Detroit. In the second part of my presentation, I will divide it into three subsections. First, I will talk about how we determine the pathogen targets, and then I will talk about the method development and optimization, and then I will finally talk about the results and the public health applications. And to start, I want to highlight one of the papers from the RCN networks by Dr. Sarah Philo. This paper came out earlier this year. Basically, it highlights the challenges and research gaps using wastewater surveillance to monitor bacterial disease. So in the paper, it highlights four challenges in bacterial wastewater surveillance. The first one is to how we're going to choose the new targets. Second is how we're going to relate wastewater surveillance data of bacteria to human disease incidence and prevalence. And then what methods we can develop to target those bacterial disease and how we're going to normalize the data. And I think this is a very important paper and outline the challenges and our research kind of trying to answer some of the questions proposed in this paper. First, I want to introduce one of the systems that we developed in last year, communicable disease wastewater surveillance ranking system. So my colleagues and I look at 96 communicable diseases in the state of Michigan and the Tri-County Detroit area. These are some of the disease as examples shown here for the year between 2014 to 2021. And the idea of this system is to prioritize what could be the targets for the Detroit area to monitor. So in the system, it includes two categories of parameters, binary parameter and quantitative parameter. So each of the disease runs through the system and calculate the score. And then there are two checkpoints, whether the disease can be detected in wastewater or human excrement. That's the first checkpoint. And then whether the disease is associated with a single pathogen or multiple pathogens. And finally, we'll have a score for each of the 96 communicable disease. And if we look at the top 30 communicable disease in Detroit, prioritized using our system, you can see that more than 50% of the communicable disease among the top 30 are foodborne or sexually transmitted infections. And then we took two sexually transmitted infections in Detroit, chlamydia and syphilis, which rank among the top for wastewater surveillance. And we're trying to develop methods to detect these two bacteria. So a brief background about chlamydia and syphilis, both are STIs, sexually transmitted infections, and they are caused by bacteria, chlamydia trachomatis, and Treponema pallidum. And if you look at the cases for the past 6 years of chlamydia and syphilis in the United States, you can see that chlamydia caused about 1.6 million cases annually. And syphilis in 2023 has about 200,000 cases in the US. And syphilis has been dramatically increasing for about 83% for the past 6 years. In the literature, it's widely reported syphilis has diverse clinical manifestations. So often this leads to underdiagnosis. And both of these STIs remain asymptomatic mostly. And so the infected individuals do not seek testing or treatment. And both STI remain underdiagnosed and undertested due to a lot of factors. And then this slide shows we investigate the fate of chlamydia trachomatis and Treponema pallidum in the wastewater. First, both bacteria, the only natural host for both bacteria are humans, and the propagation of replication are strictly bonded to the host cells of humans. And the ATP and nutrients can only be obtained from the host cells, not from the environment. And also the temperature and pH in Detroit wastewater can provide a good environment for both bacteria to maintain with good viability. In the second subsection of the second part, I will talk about the method development and optimization. First, we did an extensive literature study to look at to concentrate and isolate bacteria targets from some either clinical samples or environmental matrices, and we decided to go with a centrifugation method based on the literature and also based on the equipment that we have in our lab. And then we optimize the ddPCR assays for both chlamydia trachomatis and Treponema pallidum. And we designed a series of tests to determine limit of blank and limit of detection of these two assays. And this is our established workflow. So we collect wastewater samples from wastewater treatment utility in Detroit and also the street manholes in selected sewer sheds. And we take the samples back and we did centrifugation, DNA extraction, and digital PCR to quantify the concentrations of both bacteria. And this is our study area. So we collect samples from one is from interceptors, 3 interceptors that covers large areas in Detroit. ONWI, NIE, and DRI are 3 interceptors that covers large areas of city of Detroit, Wayne, Macomb, and Oakland counties. And then we also collect wastewater samples from street manholes that cover smaller sewer sheds in the Detroit metro area. And so here on the left, this is a map shows the captured area by the interceptors and the locations of the 3 smaller sewer sheds. These are where we collect our wastewater samples. And moving on to the results, this slide shows the concentrations that we measured for chlamydia trachomatis and Treponema pallidum in interceptor wastewater and the selected sewer shed wastewater. And based on these concentrations, we adopted a formula which was previously published in one of the papers to back estimate norovirus cases based on the norovirus concentration in wastewater. And so based on this formula, we can use the concentrations that we measured and bacteria shedding information from literature to back estimate infections of this bacterial disease. So here, if you look at the formula, W indicates the estimated infections of the 2 disease, and cDNA indicates measured DNA concentrations of both bacteria in the wastewater. And then we have a decay parameter, bacterial decay term, and Q indicates the wastewater generated by per person, which is calculated by the total wastewater flow. Alpha is a adjustment factor, basically to evaluate the wastewater dilution caused by stormwater, industry water dilution, and other uncertainty. And in the denominator, it's a bacteria shedding terms. First, P shedding indicates the rate of positive bacteria detection in urine, and Q shedding indicates the daily volume of urine generated by individual, and C shedding indicates the shedding magnitude of DNA in the urine. And using this formula and with our measured concentrations and the bacterial shedding that we investigate from literature, we can back estimate infections of these 2 diseases. So before we do back estimation, we first validate this model using 2 methods. First is uncertainty analysis. This is a very simple method. Basically, we use a partial derivative of estimated cases with respect to each parameter and tells you the relative importance of each parameter in terms of the estimated cases. So we can see here as an example, the shedding, the positive rate of detection, bacterial detection in urine has the highest uncertainty. And then we use a more advanced method, a sensitivity analysis using multivariate sensitivity analysis in R. And then we identify the top 3 parameters of in the model that has the highest sensitivity, including the dilution, the flow, and the rate of positive bacterial detection in the urine has the most sensitive impacts on the estimated cases. And this is a very busy slide with all the data that we input into this formula. Basically, these not highlighted numbers are either the numbers that we measured or the flow data from Detroit or the other parameters that we input into the model. And the green highlighted cells indicate the changing parameter in the formula. These two terms change has a different range, so it generate different scenarios of back estimation. And the blue highlighted cells indicates range of weekly estimated cases for each sewer shed. So here I highlighted some numbers in orange, which is shown in the next slide. You can see these are three scenarios of back estimation. Minimum bacteria detection rate in urine, average bacteria detection rate in urine, and maximum bacteria detection rate in urine. And under these three scenarios, we can see all the blue bars, orange bars indicate the estimated weekly cases. And then the gray bars indicate the weekly confirmed cases. And you can see there is arrows. Arrows for each orange bar indicates the difference of the decay rate. And if you look at all the figures, we can see most back estimation scenarios, the reported cases are actually underestimated. If we look further into our result, we calculate the ratio between our estimated cases and confirmed cases. And if the ratio is higher than 1, it indicates that the cases of STIs are likely underestimated. So if you look at for the 3 interceptors, all these blue highlighted cells have the ratio between estimated cases and confirmed cases higher than 1. So they indicate that the cases of both bacterial disease are likely underestimated. And if you look closer to the average estimated cases on a weekly basis and the confirmed cases on a weekly basis for each area, except for one of the interceptor for chlamydia, the remaining sites for both bacterial disease indicate both STIs are likely underreported when we benchmark against the clinically reported cases. And then if you look at the Detroit, we take a DRI interceptor from the Detroit, from the 3 interceptors, which covers majority Detroit area. And D3 is one of the sewer sheds located in Detroit. And we found both DRI and D3 have the highest estimated cases in interceptors or selective sewer sheds. And if you look at the social economic characteristics of these two areas, both DRI and D3 are located in city of Detroit with STI, where STI infections are high, and both DRI and D3 share similar socioeconomic characteristics. For example, they both have highest population density per acre, highest poverty, and lowest average income. And this is very interesting. This is something later on I will show a few slides that will involve a social vulnerability index in our analysis. Finally, with some conclusions, so this study is one of the first bacterial wastewater surveillance to monitor chlamydia and syphilis in a large urban area in Detroit, as well as neighborhood sewer sheds. And we found that both STIs are likely underreported based on the back estimation model. And the level of concentrations potentially relate to socioeconomic characteristics because they share similar trends. And the future work, we will expand our STI targets to include gonorrhea and other bacterial disease, including Salmonella and Campylobacter. And we'll also improve the back estimation model with more dilution and shedding data. And right now, if we look closer at the syphilis cases, so on the left side, you can see syphilis. We have seen this figure before. Syphilis has been surging rapidly for the past 6 years. And if you look at the closer Detroit and state of Michigan, you can see the Tri-County Detroit area, which is the metro greater Detroit area, has have higher syphilis cases per capita comparing to the overall state of Michigan. And both trends are surging rapidly for the past 10 years. And here, this is the weekly reported cases from Michigan Department of Health and Human Services for syphilis cases. And you can see the green portion indicates the syphilis cases on a weekly basis for city of Detroit. It occupies a larger percentage for city of Tri-County Detroit area. And then the gray square area are we selected 4 months in beginning of 2024 and 4 months by end of 2024 to test the wastewater samples. And these are some example plots of the triple NEMA pallidum concentrations that we observed in interceptor wastewater. And then we consider to involve social vulnerability index in the Tri-County Detroit area. This is a very, later on I will show one graph which is very interesting. So social vulnerability index involves social economic status that we look at previously, and also includes household characteristics, racial minority status, and housing types and transportation. So if you look at the three figures on the right, for Oakland County and Macomb County, most of the areas have light colors. So it indicates a lower or smaller SVI index. And if you look at the Wayne County and City of Detroit, especially City of Detroit, most of the areas have darker colors with higher social vulnerability index. And now if we calculate the average social vulnerability index, and then we plot them together with the concentration that we measure for triple NEMA pallidum, you can see that for interceptors, DRI have in general higher concentrations of triple NEMA pallidum with higher average the SVI value, and you see that both the concentrations of triple NEMA pallidum and the SVI index share similar pattern for both interceptors and neighborhood sewer sheds. And with that, this is all my presentation. And finally, I want to acknowledge and thank my advisor, Professor Irene Zagoraki, for her guidance. And also, I want to acknowledge the Detroit Project Partnership. As I mentioned earlier, we have constantly working with Detroit Project Partnership and meeting with them and getting feedback. And this is a team effort. With that, I'm happy to take any questions you may have. Thank you.
Yeah, thank you, Dr. Zhao, for your insightful presentation. So we have some questions from the panelists and as well as from the attendees. So which gene did you target, and how many copies per genome does it have in that gene? You may have discussed it, or can you elaborate it a little bit? And did you consider targeting RNA instead of DNA?
Thank you. Yeah, that's a great question. So for syphilis, we're targeting at triple NEMA pallidum DNA polymerase chain, which is often used as a target for PCR assays to target triple NEMA pallidum. And there are actually multiple citations in our paper. If you look at our paper here, and previously people used the same assay to successfully detect the triple NEMA pallidum from using the same primers and probes with ATCC standards. Actually, the specificity is generally very high, ranging from 97% percentage to 100%. And for chlamydia trachomatis, actually chlamydia trachomatis encodes an abundant protein, which is called the major outer membrane protein. So we are targeting at OMPA gene on this membrane protein, and this is also used earlier using the same primers and probes to target this OMPA nucleotide sequence of chlamydia trachomatis in wastewater also earlier, and also in clinical samples.
Okay, so you confirmed the specificity of the assay while you sequenced the product?
No, we didn't sequence the product. So we optimized the assay from the assay that we saw in the literature, and we did an extensive literature study, and we found these assays. And actually, for chlamydia, we have two assays. For both chlamydia and syphilis, we have two primers and probes to for trial test, and finally we determine one of the each to target these two bacteria.
Okay, yeah, that's great. So how did your models account for the variability or the uncertainty in urine shedding rates?
So for urine shedding rate, Actually, so for the model, I want to mention here. So actually, the model was widely adopted in published studies. It was originally proposed, I believe, from this study, back estimation of norovirus infection, and for the shedding, among all these papers, it is commonly that the shedding parameter we use is from the literature, and for us, we did a extensive literature study to look at the shedding rate for both chlamydia trachomatis and treponema pallidum in urine and in other bodily fluids, and then we have a range of the shedding rates. For syphilis, it's about 10% to 47% in different type of bodily fluids, and I believe there is another number for syphilis, for chlamydia. So it's from literature, and we have a range. So that's why we involve different scenarios of back estimation in the previous slides you can see, and we also involve uncertainty analysis and sensitivity analysis to evaluate the uncertainty of these parameters. I hope that answered the question.
So does your backward estimation models work for different mutations?
We haven't tested that, but I think that's a very interesting question. And I think that's certainly one of the areas we should work towards.
Okay. All right. So yeah, another one is like what approach did you use to identify smaller sewer shed in your analysis?
Can you repeat the first part of the question? What was that?
Yeah, so how did you identify the smaller sewer sheds?
Oh, okay. So for the smaller sewer shed, we actually earlier during the COVID-19 pandemic, we have 9 small sewer sheds in the Tri-County Detroit area with very diverse social demographic characteristics. And then later on, we optimized. We found the 3 smaller sewer sheds in Tri-County Detroit area, which are located in 3 respective interceptors, have a very good representations of different social demographic characteristics. That's why we select these 3 sewer sheds, and these 3 sewer sheds are in our routine monitoring program. So we collect samples from there on a weekly basis.
Yeah, so we have another question on like sampling strategies. Can you elaborate on how the choice between graph samples and composite sampling influenced your results?
We use the 1 liter 24-hour composite samples for our testing. We haven't used graph sample, but I believe composite sample has better representations of the wastewater comparing to graph sample. But it is interesting in the future that we can also include graph sample to compare the difference between the two type of samplings.
Okay, yeah. So I would go back to your back calculating for your model. So how did your model for back calculating cases account for variations in urine shading across different stages of syphilis, such as lower shading in neurosyphilis compared to primary syphilis?
Yeah, that's a wonderful question. So the shedding rates we looked at actually spanning a range of different stages of syphilis, the primary, secondary, and latent. Those are all reported in the literature. So as I just mentioned earlier, it's about 10.1% to 47% for different type of different stages of syphilis, and for different type of clinical samples of human bodily fluids. So the shedding rates we input into our model represent as much as we can to represent the entire stages, different stages of syphilis. But for us, at this moment, it's hard to distinguish between different stages of syphilis and different in terms of the shedding rates. So we try to include all the literature that we found for different shedding rates for different stages of syphilis in our model.
Yeah, thank you, Dr. Zhao. So now I will give it to Dr. Vim.
Okay, thank you so much. It's time for our next talk by Eric Boerwinkle and Anthony Anthony Maresso, who is missing from his screen, but we won't worry about that, I suppose. Oh, there he's back. Yay. I'll let you guys go ahead. You're muted.
Great. Thank you very much, Alec. We appreciate the invitation. Dr. Maresso and I are going to tag team this a little bit. I'll start and then lateral it to him for a lot of the primary wastewater work. This first slide really serves 2 points. One, it's always good to have a little humility and understand where the the roots of the science that we're all practicing in the case of this particular webinar. The guy on the left is Dr. Joseph Melnick, a very professorial looking guy in this photograph, who really was one of the founders of wastewater science. And this picture is in the late 50s and early 60s, where Dr. Melnick was tracking polio in wastewater in the greater Houston area and elsewhere, by the way. And then if you turn the clock ahead, in our particular case, April of 2020, the Houston Health Department, in collaboration with Dr. Maresso and Dr. Petrosino, really pioneered some of the early applications for COVID-19 in wastewater. But then the second purpose really, whoops, wait a minute. The second purpose is also, even in these two examples, it shows the variety of uses of these technologies. In the case of polio, it was really to investigate a very targeted outbreak and understand its origins, understand the sort of dynamics of coming up and then coming down. In the case of wastewater, and we're going to spend a little bit of time, Anthony and I, in this tag team, of understanding sort of the relationship of what we're seeing in wastewater with what we're seeing in terms of cases, either cases reported by public health in terms of this slide, which is the blue down at the bottom. I'll just point out one of them is test positivity rate versus viral load in wastewater. Try to understand that relationship in a variety of ways. And so, but now as we think about where the field is going and how do we get ready for a next outbreak, whatever it is. And in fact, we need to consider the possibility that it's a new organism, either from naturally occurring processes or knock on wood, I hope not, nefarious sort of means. And so we need to make sure we not only have the epidemiology side of this right, but we need to think about pushing ourselves to consider technologies that would allow us to see new viruses for a so-called disease X. And in order to do that, really, we've all of us put our heads together and thinking about how would you isolate out of a complex mixture and then understand what's in there. And in our case, probably because of our roots, it's going to be sequence based. And so really using something that looks kind of like capture, but it doesn't have to be at this point in time, of pulling out sequences. And then we're very fortunate, both Dr. Maresso and I have really a lot of background in our own training and in our labs of informatics and able to sort of parse out various types of organisms or various parts of the genome and then quantitate and understand what we're seeing. So that was really our goal in putting our heads together that will lead us to this, throughout this presentation. So I have the honor really of moving through the 87th Texas legislature something we formed the Texas Epidemic Public Health Institute. And really it is a state agency attached to higher education. And I'll talk a little bit about its various pillars and components, but our wastewater program, although it had its origins outside of TEFI, right now the program which Anthony will introduce and talk a lot about of TexWeb is an important part of TEFI and actually beyond TEFI, frankly. So the mission is to keep Texans safe and the economy strong by strengthening the capacity and resilience of Texas communities to respond to infectious disease outbreaks. And I'll point out is, I don't need to remind this audience, Texas is a conservative state. It's a business-friendly state. And so it's kind of it's a good challenge for me, is really how do you think about public health in terms of keeping our economy strong? Large businesses need a healthy workforce. Large businesses need an educated workforce. And so we're sort of cloaking public health, which has not always had the friendliest relationship with business, as we're making sure that public health becomes an ally of business. The person shown here is another partner of Anthony and I, is Jennifer Deegan, who really has helped us a lot as our liaison, both with the Texas legislature and also local and state public health. One example, I picked on this example on purpose, is one business that we're working very closely with is agriculture. Texas is an agriculture intensive state, fruits and vegetables in the south, beef cattle throughout the state, dairy and poultry in the north of the state. But I also think it's important in this slide to remind us there's a lot of water reuse in fruit and vegetable agriculture. And I think it's another application for the technologies we're applying in wastewater to water reuse and help us understand how we can keep track of various viral and bacterial pathogens in a water reuse situation. TEFI also has a pillar of training. It's embedded within a school of public health. And so I'm not going to go through this in any detail. We have both degree and certificates and mini courses. The TEFI Talks in the middle on the right is actually very specifically a business to business platform, particularly supply chains. How are businesses hardening their supply chains given their not so good experience during the early days of COVID-19? That's actually been a very successful component of TEFI. This really shows you sort of the three pillars, and I'm going to really focus primarily on the early detection part of it. There's communications. Really, I would say public health, and frankly, all of us did a very poor job in the early days of COVID-19. And so we're in collaboration with the Moody School of Communications at the University of Texas at Austin, really inventing a new field of public health communications. There is a huge area of medical communications, but not really public health communications. I already touched on training, and then early detection is our third pillar. And going from sort of right to left, and is we manage all of the claims for the state of Texas. So all health claims and insurance claims come to the School of Public Health, where we monitor the health of the population. We also monitor how we're spending our money for healthcare. The middle pillar is really bringing together electronic health records. Both Anthony and I are embedded in the world's largest medical center, the Texas Medical Center. And so I'll talk a little bit about how we're doing that for both types of symptom surveillance. And then the third, and Anthony will cover, is our wastewater epidemiology program. But I do want to talk a little bit more about electronic health record data, because it is important that we don't do, I think, wastewater surveillance in a vacuum, that we make sure we have an ability to relate it to what's happening in healthcare, whether it's in the community or in a quote unquote hospital setting. Most of you in your state or in a region have something called a health information exchange. And it's a way that healthcare uses your medical history to treat you better today. So for example, in looking out my window, if you've had, let's say, breast cancer and you were treated at MD Anderson, and then a year later your heart started going pitter patter and you went over to, let's say, Methodist Hospital, your cardiologist could bring your records over of how your cancer was treated to help understand what could make your heart be, not doing what it's supposed to be doing. And so if you can do that for one patient, we've really put in place the technology to do this with tens and hundreds of thousands, if not millions of individuals. And that's allowed us then to look at really the health of about 19 million people in Southeast Texas that we monitor literally on a weekly basis. I'm not going to go through this. This is the work of Michaela Sandoval, really of taking it, it's really putting in place a symptom surveillance, in this case, pneumonia. So we now monitor pneumonia cases in all throughout Southeast Texas on a weekly basis, of bringing the records together, having a clear definition of what we're looking at, making sure a critical step is to making sure you have something called a master patient index. There's probably more than 1,000 Maria Hernandez's in Southeast Texas with the same birthday, by the way. So that is not a good way to identify an individual. So basically a unique individual. So basically it's putting all these data together to generate a plot such as this. These are our pneumonia cases, or this one happens to be respiratory infections. And you can see in the red, pneumonia, and in the green, acute bronchiolitis, primarily, by the way, in children. And date is across the x-axis, and then some metric of counts on the y-axis. And now what we're doing is having these data, having claims data, and wastewater data. It allows us for a number of conditions, by the way. I'm just showing you respiratory infections on this particular slide, but we do a number of conditions so we can relate our wastewater data to what we're seeing in the hospital setting. So I think this is my last slide, is it's very, very important that, the relationship between our wastewater monitoring program, or TexWeb, that Anthony's going to introduce, really, and how we relate the information we're seeing both to first responders, for example, to water utilities. We have a very close relationship with the water utilities throughout the state, led by John Ballou, who's the head of the El Paso water utility. We also do a lot of interactions with our elected officials in local communities and in Austin. Elected officials don't like to wake up in the morning and see in the newspaper that there's an outbreak of, pick your disease in their community. So we make sure we have an action plan that's been put together by Jennifer Deegan. We have very specific protocols that when we identify something such as H5N1 or oral poosh, we know what we're doing in terms of how we unfold that information, both to elected officials, to public health, including the CDC, and to community leaders. So I'm going to turn it over to Dr. Maresso, who's going to talk in more detail about TexWeb. So Anthony, you want to take it over?
Sure. Allie, by my count, I have about 8 minutes to do this, so I'll probably go a little fast, but maybe okay. Eric's going to run the slides. Eric, if you could just advance to, so it has the axes. Yeah, we have some animation. Thanks, Eric. So Eric did a really wonderful job of sort of giving us a little bit of vision of where we're going, right? So imagine the health record data. You have all these 18 million records, right? Now, if you have number of SARS cases, then you can just simply, look for SARS in wastewater and correlate it to SARS cases. But what about everything else in those 18 million record sets, right? And so the only real way to get at this is to do pan sequencing. And so that's the idea here. is that we are trying to apply high throughput, cost-effective, agnostic sequencing to wastewater to give you everything, right? And so we're just going to focus on the Virome today. But one of the questions we often get addressed, like sequencing, it just gets sort of, it's great for specificity, but is it really good for quantification? It's absolutely wonderful for quantification because the number of reads you get off whatever sequencing platform that you use is not only highly quantitative, has a huge dynamic range, but it also is pretty quantitatively correlative to the viral levels, right? So that's just the idea. Okay, Eric. And it's all in a single reaction. Okay, so the approach is really to take this complicated material, you know, has all these microbes, biologicals, whatever, and get it down to nucleic acid. That's the key. Nucleic acid can be universally sort of interrogated through, you know, techniques like sequencing or PCR that are known to everybody. Okay, you could hit the button. And so we have, because we were concerned that people would kind of call us out with just using sequencing as a method, we do a targeted approach with PCR as a part of our pipeline. And so we have 14 viruses on our panel there. But the real strength of the program is our agnostic approach. And so in this particular set, we can get up to 3,500 viruses, that disease X that Eric mentioned in the data. And so that's nice that they're orthogonal, and it's nice too that we can kind of combine those two data sets together. But this is the program. We named it TexWeb, Texas Wastewater Environmental Biomonitoring. So the sites that are in the network are shown overlaid there in the map of the state of Texas. So we have 15, I believe, cities and 30 sites. So there's some cities have multiple sites. And if you count the number of people that are in these sewer sheds, we cover about 24% of the Texas population. That's pretty good. I think that amounts to around 7 or 8 million people. As I mentioned, we process the samples and we apply an agnostic sequencing approach. It's really based off of a hybrid capture technology that was developed largely through a number of papers, but came out of the Broad Institute that has actually been commercialized. So the key is that these probes have been developed against every known mammalian or plant virus. These probes are essentially pull down nucleic acid, and then we just amplify and sequence off that. And there really isn't a virus that sort of has not been reported in wastewater or that has been reported that we don't see. And we're seeing hundreds of viruses that haven't been reported in wastewater as well. And so we make this report, and this report then goes to our public health stakeholders, mostly public health department that are in the network. And it is a report that's based almost entirely on sequencing data. And if you're interested in TexWeb, you can go to the website there and read a little about it. Okay, next slide. All right, so here's a report. I'm not going to spend a lot of time into it, but it really is unique. It's not, you can't create a dashboard very simply with sequencing data. So there's a lot of components to this, but just imagine that you have the ability to get down to an actual strain or quasi-species level, and even down to a segment of a virus. That's what our report allows you to do. All right, next slide. So I mentioned we can get a lot of different viruses. I'm just giving you a little snippet of the viruses we see here. So I circled some of the important ones. Mpox, we knew Mpox was a problem well before it was being reported. That's one of the values of this, is we see things that if you don't have a directed primer set for, they're likely to come up. We see every type of flu, human metapneumovirus, all the RSVs, SARS-CoV-2 plus variants, and a lot more. All right, next slide. One of the questions we get asked often is, does the data, the sequencing data, correlate with the case positivity rate? And the answer is, whenever we have case positivity rate data, it does, the sequencing data too. So this is just an example for influenza. You can hit the forward arrow and there's your correlation. The correlations tend to be high. You can go forward. SARS-CoV-2, and there's the correlation next to it. As well as Mpox. So these are the ones where we have case data. But to Eric's point, right, we have now all the claims data for a lot of the other viruses. So you can imagine the types of analyses that we're going to do in the future. Okay, next slide. Okay, so one of the highlights is we can detect things that people don't expect, right? Or even variants that are sort of evolving in time. So this is just to show you that we were one of the first groups to detect H5N1. I know Verily, they also were amazing in being able to see this with some of their PCR specific probe sets. So we kind of reported this at about the same time, but in one of the sites in Texas, we saw avian flu and wastewater, and then very quickly within 3 weeks, it was at most of our sites. Okay, next slide. One of the cool things you could do, right, with sequencing data is you can parse out the different variants, right, or the different subtypes or clades or whatever. And so here's flu seroprevalence. So you could see we were monitoring normal flu H1N1, H3N2 through the flu season, and then boom, the red here is H5N1 picking up at the beginning of April, end of March, and of course, being very concerning and dominating in the wastewater signal, the serotype composition. Next slide. The advantage of sequencing is you can look at mutations. And so here in this slide, what we're doing is we're showing you in the red all the mutations that are novel in a wastewater for H5N1 compared to all the other sequencing data sets out there. So human cases, avian flu found in milk, and then avian flu found in animals. And one of the cool things about the wastewater, or at least our program, is we can monitor the evolution. We see novel mutations in a lot of these viruses that have never been reported in the literature. And so I just highlighted a few here for avian flu that we saw in the literature, that we saw in the data. And then in the bottom there, I'm showing you that we can map this onto known genes for H5N1. And actually, some of these are very concerning. They're either found in antibody binding sites or sites that are associated with drug resistance. Okay, next slide. We can give you the species of the viruses or the virome per city. So this is just a Houston versus El Paso comparison. It's kind of cool that each city has its own virome, right? We really didn't expect that going in, but you can identify a city by its virome. Next slide. And then the data is spatially and temporally organized. And I'm fascinated by this. We treat this data like RNA-seq data. So you could break it up by certain resolution scales and take a deeper look at that. Next slide. And so here's one of the things that we're getting into now is like what viruses positively correlate with each other and what viruses negatively correlate with each other, almost as if they move in and out of the human as a reservoir at specific times during the year. And we absolutely see that. So what is just shown here is all the negative correlations. Maybe they're blocking each other, but there are thousands of positive correlations too, and we're looking at that. And we're actually using machine learning to try to tease that apart because it's much too difficult to handle just using simple algorithms we develop. Next slide. Monitor viral evolution. So I could tell you that every city has its own viral evolution index. The actual evolution is local. So on the top is Houston, and you could just arrow it through because I think there's animation error. Yeah, and then just do 2 more. Yep. So you can see, just go back once more. Yeah, you can see here that this is an adenovirus caused gastroenteritis in children. In Houston, the mutations and the evolution we're seeing through time, highlighted in a red box, is very different than El Paso, which is in the bottom blue box below. So it's kind of cool. We can monitor the virus is changing in real time in a city-specific manner. All right, next slide. Okay, so I'll close with just what's next. Lots of things we're trying to do. I think pairing this data to Eric's health data is really sort of on a high level where we're going to go. But we're also interested in taking a magnifying glass to identify new microbes. So we developed technologies to concentrate wastewater. So that's actually shown here in the far left. This is a device we invented in a lab where we think we can get about 10,000x concentrate over conventional methods. We think we're going to pull out new microbes in that way. We actually use wastewater to find new therapies. So a different type of virus, this is a phage, these kill bacteria. And we go to wastewater to find these bacterial killing phages and actually use these to treat antibiotic resistant infections.
Can I interrupt you? Because we have a lot of questions and we only have 5 minutes left. Is that okay?
Yeah, we're done. New
Okay.
tracking. We're gonna do the entire bacterium and the entire parasitome and fungal ome next too, just like we did the virus. So we'll be able to
Thank
tell
you.
you the path.
Thank
Okay,
you
that's
for
it.
the presentation. There's a lot of questions, so I don't think we're going to get through them. But one question that we have here is, can you comment on how having PCR and hybrid capture technology as part of your sequencing prep impacts the ability to derive quantitative information from the samples?
It gives us confidence that the sequencing is a great method to use as a quantitative measure. Because whenever we have PCR data and we pair the PCR data for a virus with the sequencing data, they're correlative.
Okay.
So we never really, I'll say this, oftentimes when we have PCR positive data and it is quantitative, we see the virus in the sequencing data and it's quantitative. But one of the things that's great about the sequencing is that sometimes we have PCR negative results, but we absolutely know the virus is present and at pretty good levels by our sequencing data. So we kind of use them to inform each other. But in terms of like giving us confidence that the sequencing data is quantitative and accurate, the PCR data, whenever we have it, helps us determine.
Mm-hmm. I guess it's hard to pick because there are so many questions and not very much time. But one question is, has your data been used by public health agencies yet to inform any practical actions? And a related question is, you mentioned earlier that it was hard to put sequencing data on a dashboard, and I was wondering if your group is working on ways to actually display data in a way that it could be uptaken more readily by people who are not like highly skilled at interpreting like sequencing type data. So those are
Yes,
kind of...
we have a dashboard coming out. We call it SeqBoard. It's going to be unveiled live, hopefully before Christmas, and the public will have access to it. The reason it's taken a while to build is because we wanted to be very careful in displaying the information in a way that many different people can understand at different levels. So it's going to have 3 parts: public health part that they'll understand, a part for the sequencing nerds where they can get into the data a little more deeply, and then a public part where it's just basically color coded and there's trend lines. So that'll be online very soon for everybody. And then your first question, I'm sorry, what was it again?
Oh, it was just, you know,
actions.
Public health. Yeah.
Yeah, so there's been a number of examples of this. Yes, so all the public health departments that get our data, they do take various actions depending on what we're reporting to them. So for example, in South Texas, when they get norovirus information, they do notify the schools about norovirus prevalence. In the case where we detect new pathogens like H5N1 or ROPUSH or Mpox, the state sends out a notice to the entire public health network that these, in fact, things are circulating. And then in cases where we've seen vaccine preventable diseases, there have been notifications put out about sort of increasing vaccine uptake. So sometimes we get measles detections and or other vaccine preventable diseases. And so that's helped there. And there's probably other examples in this.
Yeah, the only thing I'll add to that is, in addition to public health, healthcare also uses these data for staffing decisions.
Yeah, that's interesting. I guess there's time for one more question. There are a whole bunch of questions related to methods. I know a lot of people in this network have been trying to use these sorts of sequencing methods themselves. So detailed questions about the bioinformatics, but I suppose if you have a link to one of your manuscripts, that might be helpful to share or look at for those of you in the audience. But I have a question. This is kind of for Eric, which is, do you see a situation where like health insurance companies might be interested in wastewater data to inform some of their decision making?
Wow. So, as I said, we do have claims data, but I cannot see health insurance using these data for either pricing or coverage decisions. They would use, it's primarily healthcare as opposed to, health insurance. Now that could change over time, but in the current repertoire, health insurance, as you know, if you're following the news the last couple days, there's a lot of sensitivity about pricing and coverage decisions. I don't think they would use these data to make pricing and coverage decisions.
Okay, thank you so much. We're out of time. Thank you, everybody. Have a great weekend, and thank you to our speakers for great talks today. Thanks, everybody.
you.
Bye.